Title of my talk and the title of my dissertation was The Logic of Change, Contextual Adaptation of a Mobile Gene. And to get started, I've kind of restructured the talk since the defense a little bit. And I'm going to give kind of a longer overview, just so we know where everything's going, and then I'm not going to come back to the outline. So again, an introduction to the experimental system is where I'm going to start. I'm going to be talking about the antibiotic resistance evolution that I've used throughout all the projects I was a part of in grad school. And again, I just recently defended my PhD from the University of Washington in Seattle from Ben Kerr's laboratory. And just as a kind of introduction, plasmids are mobile genetic elements. I'm going to be talking about these a little bit more, but that means that they include coding DNA sequences for proteins, regulatory elements, other things that typical DNA and bacterial chromosomes have information for. But these plasmid molecules are much smaller, and many are mobilizable, meaning that they can actually transfer between coexisting bacterial individuals of the same or of different species. And in part two, I'm going to discuss a published study led by Olivia Kosterlitz that I'm a co-author on, where we measured the antibiotic resistance level of 32 genetic variants of a mobile gene across 3 bacterial hosts. And we did this to examine whether resistance-enhancing mutations for one species tended to correlate with others, since this would suggest that resistance evolution of the gene might occur similarly across hosts. And then in part three, I employ an experimental evolution experiment to actually generate antibiotic-resistant genetic variants in 2 microbial species in the laboratory. First, I introduce random mutations into the gene in a modified polymerase chain reaction, or PCR, and then I select for the highest level of resistance exhibited in the 2 bacterial species. I do this for multiple rounds so that I eventually end up with mutational trajectories toward high resistance endpoints of this mobile gene in 2 host species. And then in part four, I re-engineer all of those selected genotypes during that previous part. Um, and then I compete all of these plasmid genotypes in a bulk competition separately in the 2 host species. What I'm doing there is I'm evaluating the genotypic fitness in each environment in each host. And I use this information to compare under which environments resistance mutations confer a significant fitness advantage to each of the host species over the immediate genetic ancestor. And then the last experiment that I'll talk about is another bulk competition. So another bulk kind of head-to-head fitness competition. This time with a subset of the genotypes, but now with the addition of a co-culture treatment. This means that I'm measuring the fitness of each plasmid genotype in each host species in both monoculture and co-culture settings. And I'm using this to explore whether interspecific interactions affect the outcome of plasmid evolution. Then I'll kind of wrap up where things are currently in part six. During grad school, I primarily focused on describing selective dynamics, but now as I'm kind of looking forward, I want to transition into questions about whether we can use my data to better predict and control evolution within these systems. And then to conclude the talk, I just have a slide about some early ideas for how I might explore improved predictive capacity by modeling actually fitness surfaces and adaptive pathways with increasingly complex learning models. And yeah, so to begin with the experimental system, I study the evolution of resistance to beta-lactam antibiotics, which are among the oldest antibiotics, yet they actually remain one of the most prescribed class of medications today. Penicillin was the first drug to be isolated now nearly a century ago through a happy accident made by Alexander Fleming in 1928. And Fleming was a microbiologist who noticed a mold species growing on some of his old petri dishes. And he saw that the bacteria that he was actually trying to grow, which is a staph species, was couldn't survive close to the mold, which was actually a contaminant on the plate and the mold he had not set out to grow initially. But Fleming isolated this kind of bacterial killing compound from the mold. And although it took some time to be mass produced, this drug was eventually commercialized and went on to save millions of lives during World War II and the years that followed. Um, and while antibiotics were kind of once hailed as a magic bullet, foolproof fixes for bacterial infections, we soon realized the limitations of these therapies as antibiotic resistant bacteria began to spread. And Alexander Fleming actually cautioned about this in his 1945 Nobel Prize lecture, where he said it's not difficult to make microbes resistant to penicillin in the laboratory by exposing them to concentrations not sufficient to kill them. And the same thing has occasionally happened in the body. And Fleming's warning actually turned out to be more like a premonition, as we now recognize antibiotic resistance as a major public health threat. Beta-lactam resistance specifically can manifest in a variety of ways, but it's often conferred via the production of hydrolytic enzymes called beta-lactamases. So if you look on this figure, on the bottom here, I'm showing the discovery or introduction of beta-lactam medications. So that goes back to the discovery of penicillin all the way up to fifth and sixth generation cephalosporins that we're developing now. Whereas on the top, you can see the kind of advent of different beta-lactamase enzymes in the clinic, which are capable of breaking down these new medications that we're throwing out. Um, and it's kind of formed this coevolutionary arms race, as many folks have called it. We can't actually kind of evolve at the rate that these bacteria can, but we use technology to try to kind of keep up. And what happens is these beta lactamase proteins actually bind to beta lactam drugs, and they cleave what's known as the beta lactam ring, which renders this drug inactive. So what we're doing as we develop these synthetic medications is we try to make it more and more difficult for beta lactamase proteins to access that ring by adding on different groups and such. Um, but again, bacteria are able to change the shape of this protein, which can allow the enzyme to actually expand its activity against newer substrates as well. And beta lactams are said to follow what's known as a use it and lose it trend, where the more we rely on a drug for clinical use, the faster it's rendered obsolete by the evolution of resistance. And what's problematic about beta lactamases is that these genes are often encoded on mobile genetic elements like plasmids, which facilitate the spread of resistance between bacterial species. So it's pretty simple schematic, but we can see two bacterial species here represented in red and blue. Most bacterial species have a single copy of a single chromosome, which I've also shown in red and blue. And bacteria pass these chromosomes onto their offspring during reproduction in what's known as vertical gene inheritance, just like we do. But the plasmid, or excuse me, the purple molecules are mobile genetic elements, which can actually be transferred either from parent to offspring in vertical transfer or between coexisting bacterial individuals. So between red and blue, for instance, in a process known as horizontal gene transfer. And although not the most technical graphic, this comic does actually summarize this horizontal gene transfer phenomenon pretty well. Here we see a member of the antibiotic resistance passing a mobile genetic element to a naive bacterium, giving them the ability to withstand penicillin and turning them into a superbug. Um, I like to think that the gene in this comic is the one that I study, which is referred to as TEM1, and it was first isolated from a patient back in 1963 in Greece. And this version of this protein, it's a little bit older of one. It's really excellent at degrading things like penicillin, ampicillin, the drugs that essentially came before it. But it has really poor activity against some of the newer medications. Specifically, I use TEM1 because I started in a place where it's not very resistant against a third generation cephalosporin known as cefotaxime, and I see how changes are incorporated so that this enzyme can actually break down this newer medication. I will mention that one, that's kind of a handy lab trick that I use, but two, that is actually something that's been really relevant in the clinic as well. So TEM1 is what's known as the parental allele for a wide family of what are known as extended spectrum beta lactamase or ESBL genotypes. There's about 200 of them now, and many of them have distinct resistant profiles, which allow them to break down different medications due to their different structures. And the first project that I worked on in grad school explored whether the changes in the protein that improved resistance in one host species also would improve it in the other. So essentially, Albert and the resistance member were transferring this gene. Are those two individuals necessarily benefiting from the same mutation in that gene? Are they decoding this the same way in whichever environment or context they are? And to do this, to characterize this, we look at the resistance level of all possible combinations of five mutations in the TEM1 background. So over here, we're looking at an empty pie chart that represents TEM1. So all five mutations are absent. Um, that could mutate in any one of any five ways, which gives you kind of five single mutations, one mutation displaced from 10-1. There are 10 double mutants, 10 triple, five quadruple, and a single kind of full mutation version. And for the audience, I recognize that it can be difficult to keep track of mutations, but a useful metaphor that I like to envision here is that this is like a network of protective strategies. In the lab and the clinic, we add drugs to the environment as a stressor to kill these bacteria. Beta-lactams specifically are blocking the synthesis of the bacterial cell wall, and they're analogous to an environmental stressor that we could face, something like UV radiation. And I, as the experimenter, dial up or down the degree of environmental stress, and I observe how these populations respond. Just like adding a sunscreen or adding a hat can mitigate harmful UVs, production of these beta-lactamase proteins lowers the concentration or the toxicity of the environment, with different protective strategies potentially having different efficacy. And lastly, these resistance mechanisms again can be transferable, like sharing sunscreen with a friend. And here, what we're doing is essentially evaluating whether sharing of these resistance mechanisms entail or confer similar benefits to each host. And starting with our, again, kind of baseline 10-1 reference allele on the left, we compare the resistance level of mutant neighbor genotypes. So kind of along each step here. Each of these edges is referring to the addition of a single mutation. Um, and the edge colors, whenever they're kind of collapsed into a single arrow like this, represent conserved phenotypic effects between species, and they point toward the more resistant genotype. And we can notice two things from this network. One, that adding mutations tends to improve resistance. So if you look moving left to right, arrows are usually pointing that direction. And secondly, the phenotypic effect is mainly conserved between the species. There are some arrows that are split, but for the most part, the edges are kind of collapsed down, signaling similar effect. I will mention, though, these edges that are split do exhibit some degree of species-specific difference. So we actually evaluated the resistance levels of all these genotypes in three bacterial species, Escherichia coli, Klebsiella pneumoniae, and Salmonella enterica. These are three pathogens that normally harbor these beta-lactamase genes. And whenever you see a split is where the mutation is beneficial in one species, but either neutral or deleterious in the other. Neutral is being shown in these dashed lines, and a deleterious step is one that goes backwards with dashed arrow. Um, and yeah, this just kind of wrap this up. This indicates that the same mutation could be selectively accessible in one host species, but not favored in the others, which might actually lead to the gene evolving differently between hosts. And so the figure that I'm showing here is the empirical genotype to phenotype mapping of all of those 32 mutational combinations from the last slide. Now we're adding a dimension though. So here height is corresponding to the resistance level of the genotype relative to the no mutation ancestor, TEM1. And there's a lot going on in this plot, but to summarize, if you glance at it, you can see that the overall structure of the landscapes looks similar. Looking a little closer, we can see why that's happening. It's because the rank order of genotypes is largely conserved. So the best type for both species is the one with all of the mutations present. Um, and when you kind of scoot backwards, the combination of the best one, two, three, or four mutations is also often the same between species. The mutations from the last slide that were exhibiting those divergent effects between hosts, those are actually really small effect sizes. So they're actually the horizontal dashed lines here that don't really be seeming to make a huge difference to the overall resistance level. And to actually explore this, we ran evolutionary simulations, and we found that with the subset of resistance strategies we evaluated, the effect of mutations on the resistance phenotype were largely conserved across species. This suggests that selection for cefotaxime resistance in this mobile gene will favor similar mutations between host species. In our evolutionary simulations, again, we found that the outcome of resistance evolution was similar when the mobile gene evolved in one host species, or if it underwent transient evolution in another species before returning to the initial host. We called this phenomenon evolutionary crowdsourcing, wherein we predict that plasmids can acquire correlated adaptive evolution across host backgrounds. And there's a lot more of this paper. I didn't really cover the simulations, but I'm here as a co-author, and this work was led by a postdoc in our lab, Olivia Kosterlitz. Um, but to actually go beyond evolutionary simulations, we wanted to select for de novo resistance trajectories actually in the laboratory. And to do that, I use a procedure known as directed evolution. Um, and what this does is actually aims to replicate the evolutionary process, but in an expedited timeline. So this kind of direct evolution procedure is a cycle between the addition of mutations into a biological molecule, followed by selection for an improved phenotype. This process usually repeats so that the selected variant becomes a template for another round, which continues until a desired phenotypic endpoint is reached. And in my experiment, I initiated 12 independent lines from TEM1 and selected for mutations that increase cefotaxime resistance. So there were 12 independent error-prone PCRs that were going on. And this error-prone polymerase chain reaction is similar to what's known as to a conventional PCR in that it makes many, many copies of a target region, but I actually update the reaction conditions so that the error rate of the polymerase is much higher than it would be normally. And what that does is creates a diverse pool of plasmid genotypes that I can then transform into populations of the two bacterial species I'm interested in, which were E. coli and K. pneumo. I dropped Salmonella from this one because the results were actually pretty similar to E. coli. Um, so instead, I just use these two bacteria and I inoculate them in a range of increasingly toxic environments. Then I isolate a single genotype from the most extreme environment exhibiting growth. And this scheme enables me to actually generate mutational trajectories toward increased antibiotic resistance and select for mutations with resistance enhancing effects. And at the end of the first round, I sampled 12 mutations from 12 populations of each species. These are represented by the rectangles and labeled with the round that the genotype appeared in during the direct evolution experiment. So all 12 of the populations of each species started out as TEM1, but the baseline resistance level of those species is different. So if you kind of follow it all the way to the bottom, that's the resistance level that they were actually selected at. Um, and you can notice that K. pneumo has a slightly lower intrinsic resistance level than E. coli does. And following the isolation of genotypes in this first round, from this kind of most extreme environment to exhibit growth, I sequenced the plasmids in order to determine the genetic change or changes that increased resistance. And the first thing that we can notice from these 12 populations is that there's highly repeatable selection of G238S. That means that a glycine amino acid at residue 238 of the protein was replaced by a serine. And I'll note that the mutation is extremely common in both clinical and laboratory evolution of TEM1 to cefotaxime. It was actually in the previous project as well. So we know that that's a pretty good protein. It's actually, or excuse me, substitution. It's actually known as a substitution that increases cefotaxime resistance the most. So it actually makes sense that it was showing up so often in my evolution experiment. Something that we didn't know about, though, was this D273E substitution. This showed up in two populations of K. pneumo, and the double mutant was selected at a higher concentration than G238S alone. This suggests that maybe this D273E increases resistance in this context, um, but I don't have information about it in E. coli or in other genetic backgrounds, where I'm also interested in understanding its effect. So to answer both of those questions, I reconstruct each mutation from the direct evolution experiment and I evaluate its effect individually in the mutation, as well as in the mutational background that occurred, and I do this in both species. To visualize this information, I'll plot these genotypes on what are known as really simple fitness landscapes or resistance landscapes in this case, because that's what's on the y-axis. And I reconstruct again each of these kind of mutations. So if G238S, D273E double mutant showed up from TEM1, there were kind of two evolutionary paths that could have gotten it there involving single mutational steps. You could have D273E occur first and then add G238S, or you can go the other way and add G238S first and then add D273E. And we can see from the bottom plot that the addition of D273E to G238S in K-pneumo is beneficial. So there's actually an increase in this quantity known as MIC, or minimum inhibitory concentration. Essentially, it's just a measure of resistance level. And adding D273E actually doubles resistance level relative to G238S for K-pneumo, but that same change doesn't have any effect in E. coli. This echoes findings from our previous project, but it now shows that mutations with these slight species-specific effects can actually be selected for in resistance evolution. And I'll note that this D273E substitution has occurred in clinical isolates before of an evolved TEM allele, but never in laboratory evolution of the gene, which until this experiment has only selected for mutations in E. coli. I'll mention that we also do see another form of kind of conditionality here, where D273E does not increase resistance relative to TEM1 in either species. This means that mutations that benefit resistance in one genetic background don't always increase resistance in others. That was also something that was shown in the landscape in the previous project, but I didn't really talk about it yet. But I proceeded with this direct evolution experiment for 3 rounds to actually sample mutational trajectories with 2 to 4 non-synonymous mutations. And we found out that E. coli started and ended more resistant than K. pneumo. And I don't expect folks to actually read all of the mutations here, but I will tell you that roughly the identity of substitutions appearing in both species is similar. But the frequencies that they're selected is where we see kind of more variation. In particular, I'll point out that H153R and M1A2T, these are two substitutions that both appeared in each of the species. In the second round, though, E. coli selected M1A2T in 8 out of 12 populations and H153R in just 2 out of 12. K. pneumo, on the other hand, selected M1A2T twice and H153R 4 times. And this is kind of raising the question of even if resistance effects are conserved, could there still be, species specific preferences or other phenotypes that are contributing to potentially different or potentially divergent evolution between host backgrounds? And to connect this back to our analogy, sunscreen and long sleeves might be comparable in terms of resistance, but you might still prefer one strategy over the other, especially because trade-offs might be, elicited with wearing long sleeves. You might overheat or something like that. And thus far, while I've been just describing the ability, to survive within an environment, I want to actually integrate into a different phenotype now, evolutionary fitness, which refers to both the ability to survive and reproduce within a setting. And to do that, I'm going to actually change the experiment that I'm running. So for bacteria that reproduce asexually, growth rate within an environment is a pretty good proxy for fitness. And really what I'm trying to estimate here is the fitness of plasmid host pairs, which I'm measuring in pooled competitions between genotypes within species. Because that'll allow me to actually understand how is the plasmid evolving in this context. And I track 3 barcoded replicates per genotype, which are then averaged to estimate genotypic fitness. And I say the word barcode because it's a short DNA sequence that uniquely identifies a plasmid genotype. So I labeled each of the genotype plasmids in triplicate with these 18 nucleotide regions that are way shorter than the gene itself. And that allows me to track this barcode in really, really high abundance and estimate the change in frequency of each barcode alongside the change in density of the culture. And that's what actually allows me to calculate a growth rate of each genotype in each species in each of the tested antibiotic environments. Because I actually inoculate them across a gradient of antibiotic, and I do this in both species. So I have these running side by side, a string of tubes for E. coli and a string of tubes for pneumo. And the kind of readout of this experiment is essentially a performance review. The two colors in this plot are corresponding to E. coli and K. pneumo. And here we're plotting mean growth rate over 3.5 hours as a function of concentration, specifically cefotaxime concentration. And uh, Again, these are growing in different tubes, but I want to mention that these comparisons still show differences in how well the plasmid genotype is growing in each host. And since these bacteria are still in like log phase or exponential phase, they're still growing at near their kind of maximal rate when I took this sample. There was some lag phase, and you might see, so as concentration increases, for instance, you see growth rate decreasing. Some of that might be an extended lag phase throughout that 3.5 hours. Some of it might be an increased death rate of these two different types. But for the most part, what we're trying to look at for this experiment is essentially a max growth rate of the plasmid genotype. And we can see that K pneumo has this kind of faster baseline growth rate. So in the absence of drug going all the way to the intercept here, we see that K pneumo does start at a higher baseline growth rate in the absence of drug, but it also crosses the zero line. Growth is inhibited at an earlier concentration relative to E. coli. Um, and I'll come back to this kind of why that resistance fitness thing is actually important. But we do see a similar kind of trade-off actually emerging between fitnesses of mutationally adjacent genotypes as well. And that's what I'm going to be comparing here. Here we're actually comparing the fitness for mutant neighbor genotypes in the same host to now estimate mutational effect. So at each one of these points, again, we're doing E. coli in red, K. pneumo in blue. Now we have two curves corresponding to different genotypes. These genotypes, though, are being competed in the exact same environment. So the growth differences that we're seeing here is G238S outperforming TEM1 in Klebsiella in the same tube. Um, and on the last slide, again, we saw this resistance fitness trade-off where E. coli seemed to have a higher intrinsic resistance level, but a lower baseline fitness value. Well, a similar trade-off emerges between genotypes within the same species. So the addition of G238S is good for resistance, but that same structural change induces destabilizing side effects within the protein. Um, and this means that the substitution, this G238S, does not confer a fitness increase in a drug-free environment. So if you go to the y-intercept, you actually see that the mutant is growing slightly slower than the TEM1 ancestor. As concentration increases, though, moving left to right in the plot, we see a crossover point where the mutant overtakes TEM1 in fitness and remains more fit than its ancestor for a couple of environments. Um, until eventually we see a high enough concentration that inhibits both types, and G238S no longer has an advantage. And to visualize the environments where mutant genotypes are more fit than their immediate ancestor, I collapsed the two fitness curves within each species from the previous slide into just a single one. And this shows the fitness difference between mutant minus ancestor taken within each environment, with the error bars representing the 95% confidence interval of the mutational effect. And those span of concentrations where mutants have a significant positive effect, I am referring to as the adaptive window. So from that last slide, we saw about three concentrations where G238S was growing significantly better than its ancestor. Those for Klebsiella occur between 0.063 and about 0.25. On the other hand, E. coli, that's shifted a little bit higher into concentration space before a significant effect is realized. And that's just because of these intrinsic resistance differences between our two species. And um, there we go. Those are the, I was a little behind on the clicks. And again, yeah, I refer to the region of concentration space where the fitness effect is significantly positive as the adaptive window. And the antibiotic environments where G238S is selectively accessible, again, kind of differs between species. It's staggered in concentration space according to intrinsic resistance differences. And I want to mention that we can do this kind of adaptive window analysis for each step that occurred. We just kind of shift what the genetic background is. So on the top plot, we're seeing the addition of the G238S substitution to the 10-1 background. Here, though, we're seeing the addition of N1A2T to the G238S background. And again, we can kind of do that to isolate just the effect of single mutations by doing these constant kind of mutant neighbor comparisons. And that's what I'm showing here in this now extended network. This network is all the mutational steps that were sampled in the direct evolution experiment in populations of E. coli and evaluating the fitness effect of each mutation in the antibiotic concentration where it was selected in both species. Or excuse me, in the selected species. So the fitness difference between G238S and TEM1, that was what was on the first plot in the previous slide. It's evaluated in the concentration where G238S was selected in E. coli during the direct evolution experiment. And we see that this mutation is associated with a significant positive effect. That's what the solid red edge is corresponding to in the selected environment, but there is not a significant effect in K pneumo within that same concentration. Significant effect, just for the key, is shown in color in all of these cases, dashed being a significant deleterious effect, solid being a significant beneficial effect, and then dashed non-colored is non-significant deleterious, dashed, or excuse me, non-colored solid is non-significant beneficial. And to visualize what's going on here, we know that G238S is beneficial to both of these species, but the specific environment seems to be differing between them. And to visualize what's going on, I'll just show kind of two representative examples that occurred on the same genetic background in E. coli. And these were both selected at 8 microgram per mil. And first in this plot, we see that the addition of E104A confers a significant increase in fitness to both species in some of the concentrations. So, there is a positive effect where the confidence intervals are no longer interacting with zero and are getting pushed up, meaning, okay, the adding E104A becomes beneficial for Klebsiella within these concentrations, for E. coli within slightly higher ones. Remember, though, we're just evaluating within 8 microgram per mil, which is where this was actually selected in E. coli. And at that point, this mutation no longer benefits Klebsiella. It's essentially gone past its window of opportunity. In the bottom plot, we see the addition of a slightly different substitution, but to the same genetic background now in both species. And this one, the benefit persists a little bit longer for K pneumo, and the benefit is pretty much the same for E. coli. That's what's causing these kind of slightly different things is whether or not that benefit is fully lasting. But in either way, or in either case, looking at the network at large, we see this trend actually replays out across several mutational steps. And this means that the same antibiotic, or the same antibiotic environment could select for resistance mutations in one species that might not be advantageous yet to the other. I'll also mention that every mutation that appeared individually during the direct evolution experiment did exhibit a significant positive effect in E. coli in the concentration it was selected. Any steps that you see that are significantly deleterious or slightly deleterious were ones that were actually a reconstruction of essentially double mutations, so things that skipped over individual steps. And that just suggests that our direct evolution experiment was selecting for adaptive resistance mutations. Um, Now, if we look at the network of mutational steps selected at concentrations in K pneumo, once again, we see that many of the mutations confer significant increases in fitness to K pneumo in the environments that they were selected, but most of those mutations are again not yet significantly beneficial to E. coli. I'll again highlight two cases to show what's going on here. This is actually showing the opposing trend to the previous slide, now that we're looking at kind of regions of antibiotic that are selective for Klebsiella. are not significantly beneficial yet for E. coli. So this is the addition of two mutations onto the G238S background. And again, in both cases, they are beneficial to each species, but where this adaptive window is in concentration space is again a little bit staggered due to intrinsic resistance differences. And specifically in this case, we see when these mutational steps are selectively accessible to K-pneumo, the environment is still too mild for the same steps to become advantageous to E. coli. And notably, I will mention, while most of the time we saw a phenotypic effect conserved between the two species and there was just kind of intrinsic resistance differences, we did also observe a case where a selected mutation had a significantly different effect between species in the selected environment. This mutational step was selected in K-pneumo at 4 microgram per mil, and it was actually one of the more resistant isolates at the end of the experiment for the species. But interestingly, E. coli could just not tolerate that same addition of the step. So E. coli can benefit if you from this E104V substitution if it gets added to a background containing M1A2T. But E. coli cannot tolerate the addition of E104V to an H153R background. It just causes this, I mean, the cells become so sick, you can actually see on a petri dish that the colony size is like much, much smaller. They're growing extremely, extremely slowly. Um, so there's something about kind of this combination of things that just breaks the functionality of the protein, which is really interesting because what these M1A2T and H153R substitutions are actually doing is they're not affecting substrate specificity. So they're not affecting how the protein binds to the drug. Rather, they're known as compensatory mutations. And those offset stability and do things that kind of mess with the broader protein phenotype. So how it folds, how much active protein there is, not necessarily how it binds to the drug. Um, and if you remember D273E from the previous diagram, H153R and M1A2T substitutions are that same way. They don't do things alone, but maybe they're offsetting for the destabilizing effect of G238S in somewhat different ways. And yeah. So going into the next one, this is the last experiment I'll talk about today. I add a co-culture fitness assay where the plasmid genotypes are inserted now into both species and competed across a range of antibiotic environments. I performed these side by side with monoculture assays, similar to the ones that I did before. But now I'm doing a different set of genotypes and for a longer duration. And I'm doing this for a longer duration so that I can actually allow social effects to accumulate. And I'll talk a little bit about that in a second. But remember that beta-lactamase proteins inactivate the drug. So over time, the level of environmental toxicity declines, kind of like how UV severity wanes throughout the day. Um, and I want to see how these bacteria are actually growing throughout an entire growth phase, not just a max growth anymore, but how much success are you having in an extended competition in each of these environments. Now against genotypes within your own species and in the co-culture against genotypes in both species. And from the kind of previous experiment, I used those same barcodes, but now I'm splitting the three barcodes for each plasmid genotype between host backgrounds. And I'm doing this so that I can track one barcode in K-pneumo only, one barcode in E. coli only, and one that appears in both species. And like in monoculture, I calculate the growth rate for genotypes via the change in barcode frequency and total culture density. But unique to this co-culture is the use of selective plates, and that is so I can culture K-pneumo only, both species, or E. coli. And I can actually quantify community composition over time. And from the sequencing data, we compare the fitness of barcoded plasmids across communities. So here we're looking at three plots of the same genotype in E. coli monoculture, K-pneumo monoculture, and co-culture. These curves look a little different from the last project, where resistant types appear to be growing faster in higher drug, but really they're growing for a longer duration. So over 20 hours, resistant types are comprising a greater proportion of high drug concentrations because of reduced competition. There's more space because those less resistant types aren't making it. Um, in both of the monocultures, though, we see the three bar-coded replicates are growing really similarly. That's because they're just replicates. Whereas in co-culture, you see a lot more spread. The growth rates of each barcode are a lot more heterogeneous, and that's because those are actually different species holding on to the plasmid in a co-culture environment and subject to things like competition. And with the barcoded plasmids of the same genotype in each species, I can track how the same plasmid genotype fares in each host in each environment. So genotypes in E. coli are consistently outcompeted in all but the highest concentrations, where genotype fitness shifts to favor the more resistant host. We see that the environment where the crossover from Klebsiella being the more fit host in blue to E. coli being the more fit host in red actually differs based on the genotype. So up here, this is the least resistant deleted beta-lactamase version. So it's just an empty vector control. Here is a double mutant, and here is a quadruple mutant. And you can see that this crossover point is essentially shifting rightwards toward higher concentrations as you get more resistant types. And again, this is all kind of correlated with the resistance of the actual genotype we're looking at. And I'll mention that only really the most resistant types can gain a foothold in high drug environments and actually gain in frequency for E. coli. Um, and yeah, so the some little bit more data, and then we'll wrap things up. So here we're looking at the actual plasmid host growth between communities. So the unconnected points here, so these dots represent fitness values of the same plasmid genotypes hosted by E. coli in red or Klebsiella in blue. The genotype is listed right here. And the line with shading represents the mean fitness and 95% confidence interval of the corresponding genotype in the E. coli or Klebsiella monoculture. So this line with the shading is how this same genotype is growing in that same species, but now in monoculture. And we're doing this to compare, how well do plasmid host combinations grow between communities? And for the representative genotypes I chose, Klebsiella harbored plasmids are growing pretty much the same between co-culture and monoculture, whereas E. coli is consistently getting outcompeted. You see that the growth rate is much lower for E. coli harbored plasmids in co-culture than they are in monoculture, and that's just because K. pneumo is outcompeting it. There's one exception to this case, which is when the concentration gets high enough, some of those really, really high resistant types in E. coli actually do slightly benefit from co-culture. And that's because Klebsiella is essentially, there's extra space because the Klebsiella types aren't surviving. And one more thing that I'll mention here is we actually see an interesting case of interspecific protection where E. coli can actually protect K. pneumo and allow it to grow in a concentration that it's not capable of growing in alone. Um, and on the right, we're showing the E. coli percentages. So E. coli starts at about 85% of the initial inoculum of about 10 to the 5 cells. By the time the whole culture saturates at about 10 to the 9, E. coli constitutes less than 10% of the majority of these concentrations. So it gets really, really dramatically outcompeted in low to intermediate drug. 16 microgram per mil, it does okay. It hangs around. But 32 microgram per mil is what I kind of call the battleground concentration because through the first 5 hours of growth, E. coli was the one that was driving most of it throughout the community. But as the drug began to clear, Klebsiella actually began to catch up. And Klebsiella monoculture was not capable of growing in 32 microgram per mil, but Klebsiella is capable of reaching a pretty high density in the presence of E. coli due to that protection. I'll note though, the presence of E. coli is not enough to save Klebsiella at 64 microgram per mil, and that essentially turns into an E. coli monoculture by the end of it. But yeah, that was kind of a lot to wrap things up, though. I want to talk about where I'm kind of planning to go from here. I'm still figuring out my next steps, but if I continue with this project, I'm interested in moving from kind of these description of evolutionary dynamics to prediction. And specifically, I want to talk about adaptation. So I've mentioned, this a little bit, but I haven't fully defined it. I call adaptation the process by which a population becomes more fit within its selective context. So in our system, we observe how host background, antibiotic concentration, and community context reshape the adaptive window for plasmid-encoded mutations. And I'll note that mutation, drift, gene flow, things that we think of as a little bit more stochastic, do also contribute to evolution. But what I'm focusing on is this kind of deterministic reweighting of genotypic strategies within a population driven by natural selection. And I believe that this selection process is actually similar to parameter updating in machine learning, where a system minimizes error or maximizes fit through feedback. And I want to use and leverage that analogy to actually model the structure of adaptation. So, um, I want to use essentially fitness and mutational accessibility as our response variables and try to understand how contexts modulate these responses. I want to use increasingly complex models. So I've done some work with splines, regularized regressions. I know your group is big into variational autoencoders. I'm really interested in that too. But kind of increasingly complex models that I hope can explore predictive capacity of fitness in novel mutational combinations, unmeasured environments, future time points, all of which I already have actually with these experiments. I only talked about the 3.5 hour data, but I actually do have time points, additional time points for both of the experiments that I talked about, as well as new antibiotic environments that weren't sequenced and measured yet. So I think that maps on to like model verification stuff quite nicely. And kind of in the end, what I want to do is I want to develop a framework and quantify how well these different models are learning selection. So how well they're imitating it, how well, which one quantify, or, when we quantify this information gain, matches what we see in the fitness values the best. And just to wrap with this last slide, I love this quote. I think that's used quite a bit, but it's such a good quote. The final one from On the Origin of Species, where Charles Darwin says, there is grandeur in this view of life. From so simple a beginning, endless forms, most beautiful and most wonderful have been and are being evolved. And I think what's really cool here is that our work shows that these adaptive possibilities aren't fixed. They're sculpted by context, by species, by environment, by ecological interaction. And adaptation is not this random process. Mutation and all these other things that contribute to adaptation are, but when we're just looking at selection, it's feedback driven, like learning itself, I think. And I think selection learns. Um, and evolution discovers not just what works, but what works here and now and together in the specific context that we're looking at. And yeah, that's it. This is my dog, Ginger. I